I work with National Biosafety Management Agency, and I'm glad to be here to make presentation on the topic genetic biocontrol, ensuring safe and secure development through biosafety, biosecurity, and biocontainment in Africa. What did not work today can still work tomorrow, because science and technology is a determinant factor. A lot have been said about genetic biocontrol since the conference began. But my own focus is to look at the biosafety, biosecurity, and biocontainment aspect, and as regards to Africa. As you all know, Africa is facing numerous challenges related to pest and disease management, which can have devastating impact on agriculture, human health, and the environment. Genetic biocontrol refers to the use of genetic engineering techniques to manage pest populations, disease, or invasive species in agriculture and public health. As Africa explores the potentials of genetic biocontrol, it is crucial to ensure that these technologies are developed and implemented safely and securely. That can be achieved by addressing the biosafety, biosecurity, and biocontainment concerns associated with genetic biocontrol techniques. We can greatly tap into the full potentials of these new and emerging technologies to improve public health via addressing so many vector-borne diseases such as malaria, dengue, fever, Zika virus, and other diseases. The innovative vector-borne technologies that has been used in the last 2 decades are genetic engineering, biological control, other innovative approaches, synthetic biology. But the focus here is on genetic engineering, which is the gene drive, and also I would like to preach on the RNA interferences. That's the other innovative approaches. Then what is genetic engineering? What is genetic biocontrol? Genetic biocontrol is the exploitation of genetics to get the organism to control itself, meaning that letting the nature to do the work for us. Although numerous technologies or strategies can be used in this content, optimal strategy depends on the biologic or the life history of such vector, and also risk tolerance, size of invasion, and objective of such too. The central dogma of biology that thus preventing protein from being made. Such is being used in the area of RNA interference. By so you can also move the gene too. So the RNA tricks the cell into destroying itself's own messenger RNA, while the RNA-induced splicing complex, which is the small double-stranded RNA molecule, guide the rings in what the messenger RNA have to claim. As such, we choose very specifically what genes to turn off. We also provide small double-stranded RNA and the target individual to do the rest of the work. Genetic biocontrol, a case study of malaria in Africa. Already the malaria case in Africa have actually shown that Africa have the largest burden of malaria over the last 2 decades. Malaria has affected so many people, but the fight is not over, because 7.6 million lives saved globally since 2000. 94% of them are in Africa. The success that have been recorded so far is as a result of the following interventions. The vector control interventions, such as the use of insecticide-treated nets, the inside residual sprays, avoidable diagnosis and case management, improved livelihood, and the use of chemicals. For that of Nigeria, the Ministry of Health of Nigeria have actually proved that most of these interventions have not really worked or have really make not much impact, and also cause a lot of economic burden for Nigeria. However, in Nigeria in 2001, there is a global world malaria report that stipulated that about 247 million cases and over 619,000 deaths due to malaria. 95 of cases and 96 of them in sub-Saharan Africa. Of that, 80% of deaths are children under 5 years. 1 child dies of malaria every minute in Africa. Then, in Nigeria precisely, we have 26.6% of all cases, which is about 76 millions, while 31% of all deaths, which is around 192,000 deaths. So this is the case of Nigeria. And Nigeria, from the global map, we also see, from the African map, you can see Nigeria has contributed about 26%, which is the malaria cases, the death which is at 1.3% cases out of other 53 countries of Africa, to tell you that the burden, the mobility and the mortality rate of malaria in Nigeria precisely is much compared to other countries. Also, the economical values of the loss we have. For the whole of Africa, we've lost over $12 billion. Then that of Nigeria, we've lost about $4.3 billion in these two. To tell you that if malaria issues have been addressed, Nigeria and Africa at large would have saved a lot of money. We would have channeled into other interventions too. The efforts are complicated, like I said. Resistant to commonly used insecticides, plasmodium resistant to commonly used drugs, mosquito changes in behaviors, people fatigued by currently available interventions, and high cost of procuring some of these interventions. However, the need for mosquito modification technologies, and some of these technologies which are commonly in the last 10 decades or 2 decades, as it may be, is the sterile insect technique, Ubacha modified mosquitoes, the GM modified sterile mosquitoes, and the gene drive modified mosquitoes. Much focus will be given towards the gene drive modified mosquitoes. The developmental stage of gene drive system have all begun since 1968, as you can rightly see. But the use of the CRISPR has actually been given much attention, and scientists have actually done a lot of work over the years. So there's a lot of progress over the years. Like you can see in the 2020, the antiparasite gene drive in Gambia species gene drive, rescue systems, a confined gene drive systems in anthrax. So you see all these data are showing us that work have been going on before now. This is the CRISPR-Cas gene drive, which is being used, which is being currently advocated for mosquito. So the mechanism of such operation is via Mendelian versus gene drive inheritance. As you can see, this is the Y species this is the Y type, and this is the mutation. That's the black is the Y type, and the red is the mutations. In the Mendelian's law, what happens is that there are chances of having 50% chance of inheriting the modified genes. But in the use of the genes you have chances of having nearly 100% of offspring being inherited to the modified genes. So the method of such is two, either for population suppression or for population replacement, as you can see depicted from the figure over there and here too. The pathway for malaria vector control via gene drive. First is the lab testing. Second is the confined small-scale field release, open small-scale field release, large-scale field release. Then for the lab testing, what we need to do is to assess the safety. Transgenic lines bearing these gene constructs produced and tested in small cages inside the lab. How efficient are such procedures? Then the confined small seed carriers, you need to have physical confined trials. It should be physically confined, geographically isolated to assess the etymological outcomes. What could possibly come out at the rate of that stage? So this is just the various stage how gene drive is being used to drive for malaria, the use of gene drive to drive for malaria control. So, but the whole target of it is to eliminate malaria by 2030. Small-scale laboratory studies for efficacy and safety testing under appropriate containment conditions and operating procedures are required for the lab testing. Then for the confined small-scale field release, open small-scale field release, we need confined testings under a more natural setting, but under conditions that limit release into the environment. While for the large scale, what we need is a large-scale releases in the environment to study epidemiological efficacy of the release mosquito. The biosafety concerns in genetic biocontrol. We have a lot of definition on the biosafeties, but because of this presentation, I would like to define biosafety as a policies and practices that minimize the risk of unintended consequences from the use of genetically modified organisms. The key consideration here is the risk assessment, the regulatory framework, and the public engagement. On the risk assessment, we also need to understand that before we conduct the risk assessment, you need to know who is doing the work. You also need to know the level of risk assessment. You also need to know the risk assessment should be risk-benefit assessments, whereby such risk will be reduced and benefit will be enhanced. So conducting thorough assessment to evaluate the potential risk to human, potential risk to biodiversity and the environment. This includes understanding the ecological impact of releasing the genetically modified mosquito into the wild environment. So when that is properly done, the concerns will be eradicated and the people will also buy into the technology. The other point again is regulatory framework. We need to have a robust regulatory framework. It has been observed over the years that most of the African regulatory agencies or the national competent authority that have such mandate have not really looked at incorporating this into their own systems. Now that would be a growing problem because nobody will accept, or the people will not going to accept, since there is no only plans or guides on how to regulate such. So we need a robust regulatory framework that govern the research, development, and deployment of this technology. This includes guidelines of field trials and commercial release, public engagement, involving local communities, stakeholders, discussions about genetic biocontrols. This public engagement is all about the buy-in of the people at the top, but look at everybody have right, or everybody need to be engaged on that too. The biosecurity concerns, we talked about the containment protocols, the monitoring and surveillance, the emergency response plans, whenever there is such release. Then the nexus between this is that we need to look at these factors. We look at the biosafety issues, we also looked at the biosecurity issues too. But the common factor there is in between this, which is this part here, which is the access control, personal management, inventory of biological hazard, proper decontamination, proper shipping and practice and procedures. All this come up with both the biosafety and biosecurity measure. When it's being put in place and when it is being fully enacted, you'll find out that people will now buy into this technology. The biocontainment, genetic safeguard, designing organisms with genetic traits that limit their ability to reproduce or survive outside the controlled environment, e.g. using gene drives that are self-limiting. Also physical containment, utilizing the physical barriers and control environments, such as greenhouse or laboratories, to prevent the escape of the GMOs during research and development. Then the most important thing, the post-release monitoring. We need to implement monitoring programs to track the behavior and the impact of release organisms, ensuring that any unexpected outcomes can be managed effectively. As I conclude, the development of genetic biocontrol technology in Africa holds great promise for addressing malaria challenges and other public health issues. However, ensuring their safe and secure development requires a multifaceted approach that integrates biosafety, biosecurity, and biocontainment measures. By prioritizing these aspects, African nations can harness the benefits of genetic biocontrol while safeguarding human health and the environment. We will honestly need collaboration among government, researchers, industry stakeholders, local communities that will be essential to build trust and ensure the responsible use of these innovative technologies. Thank you.